Okay, so welcome everybody to today's edition of the Corking Seminar. Our speaker is Andrew Brouwer, and he'll talk about identifiability and model reduction of pharmacokinetic models of carbon-stable isotope breath tests. All right, please, Andrew.
Thank you so much, and thank you for the invitation. So this is going to be a very applied talk. Although my PhD is in applied mathematics, I'm in an epidemiology department at the moment and spend most of my time thinking about kind of real world data and applications. So although there won't be too much theory in this talk, hopefully it will give you a sense of the important real world outcomes of the theory that you're all working on. So before I get too far into this, I just want to acknowledge my collaborators, including Douglas Morrison, who does a lot of the experimental aspects of this, and my colleague Gwen Lee at Rutgers, who has been a great collaborator. The work is funded by the International Atomic Energy Agency, as well as the NSF. So what I'm going to talk about today, start with looking at gut function disorders, and in particular, environmental enteric dysfunction, a specific kind of gut dysfunction. But we're going to kind of move in to talk about the carbon-13 sucrose breath test and develop a pharmacokinetic model for that test, and kind of really spend a lot of time thinking about identifiability and model reduction, and ultimately hope to develop a model-based diagnostic. You may say, where's the math in this? But a lot of it kind of comes into the aspect of understanding the identifiability and model reduction for this model. So where I want to start with is thinking about gut function disorders. And gut function disorders really have a large global burden. Chief among these is something called environmental enteric dysfunction, or EED. And this EED is related to what we call chronic enteropathogen exposure. So meaning pathogens that cause diarrheal disease and kind of being repeatedly infected by them. And this is caused by kind of lack of access to improved water, hygiene, and sanitation. And it's thought to be basically ubiquitous among about the 2 million people that don't have access. And on the other hand, we have things like celiac disease, which is an autoimmune enteropathy meaning a disease of the guts, and that's related to gluten consumption. It's probably much more familiar to this audience, and this happens in genetically susceptible people. And that's estimated to affect about 1% of the global population, but of course 1% of however many billion people we are on the globe now is still a very large number. And gut function disorders, and these two in particular, have kind of characterized by similar kind of syndromes of symptoms. And these can include things like villus blunting. So in the small intestine, you have all these finger-like villi that are absorbing nutrients from the small intestine. And so what happens in these syndromes is that the villi get blunted, so they become flatter, as we can see kind of going from left to right in this figure. And they become more permeable, meaning that they're letting in to the bloodstream things that probably shouldn't be in the bloodstream, like bacteria or molecules that are too large. And this also kind of comes along with kind of chronic inflammation as well. And so this constellation of traits we typically see. And what's particularly concerning is that in children, these gut function disorders can result in both malnutrition due to poor nutrient absorption and what we call stunting. Stunting meaning they're too short for their age by 2 or more standard deviations below the average for their population. And this impacts about 150 million children worldwide. And stunting is really a problem because stunted children tend to have poor cognitive outcomes, and that leads to having lower earning potential and living in poverty. somewhat counterintuitively, they're also more likely to suffer from obesity and kinds of chronic disease, as well as to have stunted children. And so that you can see the kind of the perpetuating of the cycle here, with that stunting in children being both due to kind of like epigenetic factors that are inherited and kind of the social outcomes as well. And so what we'd really like to be able to do is determine who has EED and provide interventions that are going to hopefully stop the interpathogens, repair the damage, increase nutrient absorption. But once the children are stunted, there's a little bit of like too little too late in terms of the interventions that kind of are practically able to do. But testing for these diseases are actually pretty challenging. The gold standard is an intestinal biopsy. And as you can imagine, doing intestinal biopsies is impractical, particularly in low resource settings, particularly not clear that it's kind of even ethical to do. Are the harms outweighing the benefits? Especially for young children in low resource settings. There are also other tests, such as dual sugar tests, where you give a small sugar and a large sugar, and you see like the ratio of them in the urine. And so if somebody doesn't have gut dysfunction, then the small sugar should come out in the urine and the large sugar should not, because it's being filtered out by the villi. But if you have permeability, then you might have the ratio closer to 1. The problem is that although these tests work theoretically, basically everyone who does them kind of gets different answers. And so they're really inconsistent across platforms and not a good test either. And so what has been proposed and kind of championed by the International Atomic Energy Agency is something called stable isotope breath tests. And with a stable isotope breath test, somebody is given a small amount of what we call a tracer, some molecule that has a carbon-13 or perhaps a different stable isotope. These stable isotopes are not radioactive. There's no kind of functional difference from the body's perspective. And they are digested, metabolized by the liver, and come through the blood and exhaled on the breath as stable isotope carbon dioxide. And so what's nice about these tests is that they have a really great potential. They can be done non-invasively in low resource settings. Basically, children are just kind of breathing into a tube or a bag that is then capped and sent for analysis. The analysis, in theory, can be done in the field as well with a kind of a mobile spectrometer, although kind of practically today they're kind of sent to labs. And one tracer that has been kind of proposed is sucrose or table sugar. And the reason for this is that the enzyme that's responsible for breaking down sucrose, called sucrase isomaltase, it's a glucosidase enzyme, meaning it breaks down starches and sugars. It's secreted at the tips of the villi. And so it's hypothesized that if the villi are blunted, that's going to disrupt the activity of this enzyme. And there's some preliminary evidence of this for both EED and celiac disease. And so what's going to happen here is that you give someone sucrose. That sucrose is tagged with carbon-13. That enzyme will break it down if you don't have gut dysfunction. And if you do have gut dysfunction, it's going to not break it down as well or as quickly. Let's see. The question, though, is what do we do with the outcome data? So if you are doing this test, you get serial measurements of carbon-13 carbon dioxide, and these produce what we call a breath curve. For all practical purposes, we're not going to worry too much about how we get from the actual concentrations in the data. What we're actually going to work with is something called the percent dose recovery rate. So what fraction of the dose that you gave is being recovered instantaneously at any point in time. And if you were to integrate this curve, it could be as big as 1, although it will not necessarily, you'll not necessarily recover the whole dose because it can also be excreted in urine. And so here I have a set of breath points and a curve that I've just fit with empirical curve fitting. However, it's really unclear what aspects of this curve tell us anything, if anything, about gut dysfunction. So there have been a number of summary measures that have been proposed. These things like time to peak, or what the peak dose recovery rate is, how much dose has been recovered by 90 minutes, how much time it takes to recover 50% of the dose, and so on and so forth. But it's not clear that any of these tell us what we want to know. And so the goal here is to use pharmacokinetic modeling to connect the breath curve to the underlying biological processes. So we're going to use differential equations to describe the transport of the body, transport of the tracer through the body as a set of a function of biological parameters that actually mean something. And so what we need to do is confirm that these biological parameters determine a unique breath curve. That's identifiability. That's where our differential algebra comes into play. And we need to know that for real breath curve data that we can actually find a best fit set of uniquely identifiable biological parameters. And so big picture, we're taking a breath curve, seeing it through the lens of this kinetic model and making inference ideally about sucrase activity. And so we have this conceptual model as a starting point. We know that we've given a tracer that goes into the stomach. Fortunately, it's possible to design tracers that can bypass the stomach pretty quickly. Otherwise, the stomach is like kind of a huge pain from a modeling perspective, because depending on how much goes into it, it can like sit in there for a while. So what we really want to do is have a small liquid tracer. That then goes into the small intestine. The enzymes are going to break down into the moieties of glucose and fructose, and be processed by the liver into bicarbonate. And bicarbonate can be what we call sequestered in a fast pool and a slow pool. The slow pool is adipose tissue. I've been making the joke that I can never quite remember what the fast pool represents biologically. And so every time I go to Google it, I'm like, fast pool bicarbonate. And I get all of these like giant bags of bicarbonate for your backyard swimming pool. And it's just kind of hard to find what it actually is. So, but that's just a joke. So and then this bicarbonate is released from the body either through urinary excretion or, as we observe it, pulmonary excretion as carbon dioxide. And our goal is to infer things about this specific rate within the larger context of the model. And so probably, I never quite know with any audience how much people are familiar with identifiability as a concept, but I'll talk us through it anyway, in case there are folks that are less familiar with it specifically. So we're interested in knowing whether any parameter in our model gives a different output. So if every parameter does give a different output, then we can see if we see a specific output, we know which parameter generated it. Okay, that's all well and good. But if we have multiple parameters that are all giving the same output, then if you observe that output, we don't know what the true underlying parameter is. And so that's what we refer to as not identifiable or unidentifiable. And so what we're going to think about here is this sort of general ordinary differential equation model setup, where you have a set of model equations. Here dot will denote derivative, and what we call a measurement equation that's connecting our equations to data. So with the x's, we don't observe, they're doing something under the hood. The y is what we are connecting to our data. So we think of it as being observed. So some state variables, time, our parameters here will denote by theta, and we have some known inputs that in this case will be how much tracer we gave to a participant. And so here's probably my one slide really on differential algebra. As you all know more than me, we're going to be thinking about our models and our measurement equation as differential polynomials. And the goal is to really reduce these equations either by something like Ritz pseudo-division using Grosvenor bases, Or in this case, what I'm going to be talking about today is really simple enough that we can just use substitution. That's really only viable in these kind of linear models. As soon as you get into non-linear models, some of my other work does, I look at transmission models for infectious disease, and those, due to the non-linearity, become very difficult very quickly to do, at least by hand. And so our goal is to find what we call the input-output equation, a non-unique equation that's a monic polynomial only in terms of y, theta, and u, and perhaps derivatives of y. U here is a constant, so we're not going to think about derivatives. And so our coefficients of the input-output equation are the structurally identifiable parameter combinations. So anything that we know we could see as a coefficient in our input-output equation is something that we could determine from perfectly observed data. And so ultimately, if the map from the parameters to coefficients is 1-to-1, then great, our parameters are identifiable.
Quick question. So Andrew,
yes.
Could you go to the previous slide? Yeah, so the last bullet might be not as intuitive for the audience. I wonder if you could say a little more while thinking of the coefficients being structured identifiable parameter combinations.
Sure. I think it'll become a little bit clearer as I go through an example, but the idea is that all of the information that we can say that we know about a system is condensed in the input-output equation. And that includes our parameters. So if we have, let's say, parameter theta1 and parameter theta2, and in our input-output equation coefficients, they never appear individually and only appear as the product theta1, theta2, then any value of theta1 could be compensated by a commensurate change in theta2, such that the product is kept the same. And so what we would say is that individually those two parameters are not structurally identifiable, but their product is. And so what we could think of is if the map from the coefficients, which contain all of the information about this parametric information about the system, is 1-to-1 with the parameters themselves, then each individual parameter can be known. But if it's not 1-to-1, as in the case of this product, then the parameters would not be identifiable. Does that help?
Yes, I think this is very helpful. So this is kind of saying that if they're identifiable, they have to show up there in a good way.
Yeah.
I wonder, what can be said about the reverse? If they show up those combinations, why we're thinking about them as identifiable?
So could you define reverse for me?
Oh yeah. So in the last bullet says that the coefficients are structurally identifiable parameter combinations. So this is saying that so you eliminate the x variables, you got just equation on the y variables, y, theta, and u, and so this is saying that the coefficients of those equations will be identifiable.
Correct.
So is there some intuition like why this is typically is the case, like why we're thinking that way? Because to connect it to your picture where you had like P1, P2, P3 and different curves, like is there some way to connect it to that picture?
Sure. Okay, so what we're saying is that if we perfectly observe the curve, then and if we write the curve as a function only of the things that we know, so y, u, and some combinations of the parameters, then if we have perfectly observed data, then I know exactly what the dynamics of that curve are. I know exactly what the coefficients of the differential equation must be to come up with that curve. I already know what y is because I have it perfectly observed. I know what u was, and thus the only things that were unknown were the parameters, the coefficients of this differential equation, But since I'm observing it perfectly, then I can be able to determine those coefficients. So that's the intuition.
Oh, thank you. So from curve, you go to its defining differential equation. And then from there, you go to these are the determined coefficients.
Exactly. So the point of the differential algebra is to get rid of any of the unknown different parts of the differential equation and write it only in terms of the known parts. Great.
Thank you.
Let's see.
I have a question I just posted on the message, but I'll
Yes.
read it. Can you clarify the type of functions f and g? Are they just polynomial, or do they involve any derivative of the x? I mean, not x, but well, perhaps x, theta and u.
Let's see. So f and g have to be rational functions. Where are we? Rational functions of x. So no, we're thinking of the system as always in terms of right, just of the variables and not of the derivatives, although you could have written maybe the original system had higher order derivatives, but you can always write them as first order, I guess, but they're rational functions. Does that answer the question?
I mean, that's in terms of theta and u as well. They are just rational functions, or do they involve derivatives of theta and u?
Well, we are thinking of them as constants, so there are no derivatives. Okay. Yeah, parameters are constants, and
all right, so
the unknown could in theory be a time varying thing, in which case we would assume that we did know the derivatives of it.
Okay, all right, thank you. So they're just basically polynomial. F and G are just polynomials then.
Correct. Yes.
Okay.
And so I think William was also asking whether we can allow derivatives of inputs in the equations. Does it ever occur, or it just does never occur in practice?
It's allowed. It's just we don't typically do it in practice, but it's allowed. There's nothing wrong with it.
I mean, I think it is allowed that you can turn some knobs and change parameters, but you don't measure them continuously. I mean, if you change parameters, it's a different problem.
Right, and so as depending on how you've specified your model,
because you said they're constants, right? So for any particular model, then they don't change.
Right. So if your parameter
if you change them, you can of course turn knobs.
Right. So if if your parameter is time varying, so like switching functions is maybe needs a different formulation that's kind of time-based. But if your parameter is not constant, then if you can write a differential equation for it, then you just add that to your equations x. So for the purposes of what I'm talking about today, the parameters are constant and the known inputs are known polynomials that in theory could have derivatives, but I will not be talking about their derivatives.
Okay,
Any other questions about this setup? Great. This is helpful. I was giving the same talk to a bunch of different audiences, and so you never quite know who's gonna have questions about what. Okay. So the way that we might approach it, at least from the data side of things, is that maybe you have a set of data and you've specified your model as, let's say, y equals m1 plus e to the m2 plus sine of m3 over error function of m4. And you might say to yourself, can I separately identify m1, m2, m3, and m4? And in this case, I think it's pretty clear that you cannot, but in general, for more complex models, it's not always clear. So we're going to go through this, given time, maybe a little bit quickly, but I can write down the set of ordinary differential equations for my pharmacokinetic model as a function of the concentrations of the isotope in each of the compartments. And I'm not going to go through the substitution, but or finding the input-output equation, you do substitution or you do pseudo-division, or find a Grosvenor basis. And what we find is that the input-output equation for this model has 4 non-constant coefficients, or excuse me, integer or non-real number coefficients. And so they are singletons, doubles, triples, and quadruples of underlying rates in the model. And what we can do is, if scrutinizing these numbers or kind of using other approaches, we can note that there's a simplification we can do here. I'm going to reparameterize this sum of row 30, 34, 35, 36, and 43 as alpha, and beta as row 43 times row 30, 35, and 36. And so if I do that, then all of a sudden my P1, P2, P3, and P4, which were coefficients of my input-output equation, become quite a bit more simplified. And what you'll notice is that row 30, 34, 35, 36, and 43, given this information alone, could not be identifiable because they don't uniquely appear in our identifiable parameter combinations. Again, remember, we're thinking of this as the map from parameters to coefficients, and we're wondering if that is injective. In this case, we solved them by substitution. If you want to do this otherwise, as I said, they can become very complicated. You can attempt substitution or its pseudo-division by hand. Alexei Software Cyan is great. It's where I usually send people. There are others like Strike Gold. And there are numerical approaches like using the Fisher information matrix or subset profiling. I'm not going to go into too much detail there, but can talk offline.
Hello, could you put up the previous slide again? Yes. So here, what you're doing with alpha and beta is what I would call aggregate variables of the parameters.
Sure.
Now, I was wondering why row 1, 2 plus row 2, 3, which appears several times in your P1, P2, P4 equation, why don't you put that as another parameter?
Because it also shows up as row 1, 2, row 2, 3. So they're different.
The addition one, I mean, not the product, but also the product. In other words, if I introduce 2 more aggregate variables, I mean, okay, gamma and delta, where gamma is row 1, 2 plus row 2, 3, and then let's say delta is row 1, 2, and row 2, 3. And of course, with these 2 parameter equations, they are basically the same because from these 2 equations of gamma and delta, you can actually solve for row 1, 2, and row 2, 3.
That's right. So I think you're anticipating this slide, I think.
Oh, okay.
Which is that if
I just wonder, at what stage do you use aggregate variables, or do you just do them in stages as you discover more and more that you can do? Because you see, when you aggregate the variables, you are basically changing the non-identifiable into identifiable by eliminating the non-identifiable, replace them by these aggregate variables.
That's right. Yes. So, there are ways to kind of do this all at once. I'm taking this approach because I think it's more intuitive for at least this presentation, right? So I was let's get rid of, through alpha and beta, all the ones that are not at all identifiable. And then, as you pointed out, for row 1, 2 and row 2, 3, it's not quite the same. We can't just get rid of them by making a smaller number of aggregate variables.
No, not smaller, but simpler, let's say.
Well, I mean, it's a matter of perspective, I think.
I mean, in other words, you leave the solving of the row 1, 2, and row 2, 3 in terms of, let's say, gamma and delta later, because you don't need to know them separately in terms of these equations that you already have.
Yeah, that's true, but there is a difference between what's there, between the previous variables in row 1, 2, and row 2, 3, which here we can see
the dimensions of are different, of course.
Right, and so
the dimension in terms of solvability.
Right, and so for row 1, 2, and row 2, 3 are locally identifiable because we can know their sum and product, meaning that they have two values, but we don't know which one is which. And so that has a different type of identifiability than those previous variables, which were not identifiable. These two are what we call locally identifiable. So there's a neighbor.
Well, of course, if you try to solve them, there is a quadratic equation involved, I guess.
Exactly, exactly.
So it is not exactly identifiable in terms of the two aggregate variables. Yeah, you're right. Because of the quadratic
Yes.
nature.
Yeah.
Okay, thank you.
Great. I have a couple more questions. Is that okay?
Sure.
Yeah. So those aggregate variables, parameters that you found, were you able to find the corresponding system of differential equations that would depend on these parameters, new parameters?
It's always possible. The question is it particularly useful?
Mhm.
In this case, yes, I at some point did write them out, but it's what's a bit challenging is that it's not always possible any earlier than going directly to the input-output equation. So it's always possible in the input-output equation, but there may or may not be kind of intermediate differential equation models that use some, reparameterization of the original x's. Yeah, so it's possible. Yeah.
Okay, thank you. And so another question, how you found them? Like, was it by visual inspection or you use something?
In this case, so what I typically do is in this case, we'll say it's pattern recognition.
No, I think in reference to Alexei's question, there is this technique called dimensional analysis in equations where you can identify aggregate variables that affect the system. So is that actually used or not? Or do you just simply let it come out of Grosvenor basis or whatever?
Yeah, I mean, I think letting them come out of Grosvenor basis is, is kind of how I've been approaching it. I'm not specifically familiar with the technique you just referenced, but I would say
the dimension analysis of, let's say, in chemical equations, kinetics, it's a well-known method.
Sure. So likely applicable here then. For the purposes of this specific example, I think kind of inspection is sufficient, but for more complicated things, things like dimensional analysis or kind of leveraging Grosvenor bases can be useful.
Thank you. Yeah.
Other questions while we're here? Chat here. Yes. Okay. However, there's also some information in the initial conditions, because if we're perfectly observing our y's, we can observe what the initial conditions of the derivatives are, and we can see that we have these additional parametric information. And altogether, if I were to put them all together, actually a minimal set of locally identifiable parameter combinations, we actually, because of the initial conditions, can recover row 30, row 34, row 43, and the only thing that we cannot is row 35 and row 36. That's a bit anticlimactic from the purposes of this talk, but we'll come back to them in a sec. So then I think to point to Alexei's question, can we find a subset of differential equations that account for that non-identifiability? What this means in this case is that we can't distinguish between the two orange arrows if we're only observing the blue arrow. And so in this case, there is a very simple reduction, which is simply removing the slow compartment and adding it to that other orange arrow, and rephrasing here row 35 plus row 36 is this row 30 minus. So it's leaving 3 and going into the ether. So this is now a structurally identifiable model, and every parameter in this model can be uniquely determined from a perfectly observed trajectory. However, we're not done yet because we do not have perfectly observed trajectories. And so now we need to take a little bit more of a statistical perspective of acknowledging that we have real data. The observed data here, we'll call them ZIs, as being based on the Ys, which are kind of the parts of the model that connect to the data, plus some measurement or observation error. And so practical identifiability is a question of whether the parameters can be uniquely determined from a given set of data. We've already said structurally that they can if we perfectly observe the trajectory, but we don't perfectly observe the trajectory. So that might mean things like the data collection are missing important dynamics, either for systemic or one-time reasons. So here, maybe all the dynamics are, really annotate here. So maybe all the dynamics are happening before the data are collected. Maybe they all happen after. So within the context of a human life forever, for example, these would only happen like 300 years in the future. Maybe you're being a good scientist and you're measuring every day at the same time, but a lot can happen in between those measurements. Or what we normally see is that your data are just too messy and like there's a lot of different curves that could reasonably fit those data. And that's the situation we have here. And so the goal here is to adjust our parameters, and we're going to use a maximum likelihood context to match the data as well as possible. What does as well as possible mean? Well, we'll define a cost function. Here, a scalar function of the parameters that assesses the goodness of fit. In practice, these cost functions use what we call the likelihood function appropriate for the data. So that might be least squares or Gaussian for normal distributed data. Maybe you want a binomial likelihood if you have a yes-no data, Poisson distributed, maybe for case data of some sort. And so probably many of you are familiar with maximum likelihood estimation, but just in case, we're thinking our cost function as being a function of the parameters, and really that it's some function of both the model at that set of parameters and the data. Really, so we're going from our parameter in our P space to a scalar C of theta as a real value. And so we define our maximum likelihood estimator as the parameter that maximizes the likelihood, or in kind of more practical terms, minimizes the negative log likelihood, which is equivalent. Because optimizers like to find minimums, not maximums. And so if we were to visualize these cost functions as a function of one or more parameters, so we have parameter 1 on the x-axis and parameter 2 on the y-axis, and the colors are the cost function. On the left, we have a single minimum. We like that. There's a clear value of both parameter 1 and parameter 2 that correspond to the minimum. In the center, maybe there are multiple minimums, some local, some global. And on the right, we have structural unidentifiability, where a whole kind of set of values, perhaps a product of parameter 1 and parameter 2, all give the same cost function. You cannot differentiate how well the model fits the data for any value along this ridge. And the thing that we like to do here are use parameter profiles. And the way that we think about this is you take one parameter and fix it at a set of values. And at each of those set of values, you find the best fit to the data, allowing all the other parameters to be optimized. And so that's saying that, like, if you fix this parameter at this one value, what's the best you could possibly do given the remaining uncertainty in the system? And so I'm not going to define it specifically here. It's a bit cumbersome, but we'll call that C star of theta I. So those might look flat here on the left. They may have different curve shapes that I'll come back to in a second. But we can define a likelihood-based confidence interval for that parameter using a chi-square distribution. Given the time, I'm not going to go into this too much, but this is quite related to the likelihood ratio test. And in general, we're going to say that a parameter is not practically identifiable if it has an infinite confidence interval. And so what that looks like here is On the left, we have structural unidentifiability. All values along the profile are such that your parameter of interest is being traded off with other parameters to continue to perfectly fit the data, or have the same best fit, rather. On the left, or on the center, we have kind of the parameter can't be too low, otherwise it doesn't fit the data. But anything that's large enough can fit the data well enough. And so this is going to have an infinite confidence interval on the right. But you'll notice that if we were to change the confidence threshold, we could actually make it not be infinite anymore. And so the idea of practically unidentifiable is dependent on the level of significance, which some people find unsatisfying, but for practical purposes is what it is. And on the right, we have our lovely likelihood well, where we have a curve with a finite confidence interval. So if I were to do this for our pharmacokinetic model, I can give the profiles. This is just for one individual, but we could do it for many individuals. What we see is that we have identical profiles for row 1, 2, and row 2, 3. That's consistent with the local identifiability we saw. They're interchangeable in some sense, or in the sense of interchangeable. But these other 4 parameters have infinite confidence intervals and are not practically identifiable. And that's not super surprising, since they really only depended on like higher level derivative information at time 0. So it's not surprising that we're maybe not able to determine them. And so generally speaking, what do you do? There's not always a right answer. For structural unidentifiability, there is a right answer, but for practical, there's not necessarily. So for Alexi's question about are there intermediate sets of differential equations, for structural, there's always at least the input-output equation, and usually there's something in intermediate. That is not necessarily the case for practical unidentifiability. One more reason that it is perhaps not as satisfying. So what we typically do is make assumptions about parameter values, such as sending it to 0 or infinity. And those of you that are interested in differential geometry, I highly recommend looking at the manifold boundary approximation method, which is closely related to this idea of practically unidentifiable. And these assumptions may or may not correspond to structural changes, as I said. And so what I'm going to do here is just kind of note that row 4.3 is practically insensitive. So it basically is not changing the fit within our level of significance. And if I look at my model, that's essentially saying that the dynamics of the fast pool can safely be ignored. So I'm just going to chop it off. And we're going to iterate this process until we have a model where all the parameters are practically identifiable. So for this model, I now have one identifiable parameter and identical profiles for the others. But what you'll notice here is that I have 3 parameters, but only 2 likelihood wells. That's a bit strange. So what's going on? So I'm not, in the interest of time, not going to show it, but you can check to see what's happening in your parameter profiles. In this case, generally speaking, the larger parameters are twinned, so that you have 1 small and 2 large parameters in those wells. This was just one individual, one set of observations, but we can do this over, in this case, we have 20, and this is what most often happens. And so that leaves us with this model where you have 3 rates that are, you know the values, but you don't know which one is which. You have one that's twinned, so you have a gamma distributed process, and one that it's on its own as an exponentially distributed process. And you can only determine pulmonary from sequestration or urinary excretion. If we get a set of profiles, we can identify each of them. Again, rho is a gamma distributed process. Pi rho describes an exponentially distributed process, and kappa here is the fraction of tracer that is exhaled. And if I were to apply these to my data, here's just 4 of them. We have 20. You'll see that the best fit full model is in black, the best fit intermediate model is in blue, and the best fit reduced model is in red. And you might see a little bit of blue peeking out here or there, but for all practical purposes, we cannot distinguish between the reduced model and any of the previous full models, suggesting that we've done our job well. This solution has a closed form solution, which is kind of nice, and we can compare it to commonly used empirical curves that are typically used to do empirical curve fitting. And you'll see that although there's some similarities here and there, it's actually a very distinct approach to doing that curve fitting, and preferable, at least in my opinion, because the parameters are interpretable in terms of the biology, unlike the other curves, which are completely empirical. But although we have this model, we haven't achieved our goal because we don't know how to make inferences about the rate of sucrose hydrolysis, which is what we care about. Which of these parameters correspond to that sucrase isomaltase activity? So experimentally, we can inhibit sucrase isomaltase. We're going to use a diabetes medication called Reducose, which is marketed as a blood sugar regulator. And what this means is that this Reducose comes in and it is blocking the sucrose from being attached to the enzyme sucrase isomaltase. And so this is an experimental inhibition that we're hoping kind of mimics this gut dysfunction. And if I were to do this experimentally, my collaborators do this, we can see that increasing doses of Reducose flatten our curves. So as I go from red to orange to this kind of sage color, the curves kind of get smushed down. So the peaks are moving a little bit to the right, generally speaking, and lower. And so the question is, how is this represented in the model parameters themselves? And if I look, primarily it's associated with rho, that first parameter that's kind of capturing that gamma distributed process. So we kind of have this decreasing values across the board. Not much change here in pi rho, and not so much in kappa. Here, this is not identifiable. Kappa is not identifiable if you don't observe the peak, because you don't really know if it's going up or coming back down. So we're not going to worry too much about that difference there. So what we'd love to have is a diagnostic threshold that distinguishes between, no inhibition and inhibition. This is a clinical diagnostic. If you were to take a test and the doctor says you have dysfunction or you don't have dysfunction. But, there's going to be some overlap, generally speaking, in terms of the values of any test. And so if you have, such a given value of a test, do we say that you have inhibition or you don't have inhibition? Let's see, question from the audience, what does pi mean in my formula? That is referring to just some factor, because pi rho is always a smaller value, pi is a value between 0 and 1. It's just, doesn't matter too much, it's just a parameterization trick that I'm using here. Okay, so if I were to pick a value of the parameter here, then this is not a really great choice because although I do have a high true negative rate, which is good, meaning that I'm not going to, if the test says no, then it's no. But if the test says yes, you don't really know if it's right or not. So that's not a great choice. Or I could pick over here and have a high true positive rate, but then if it says no, but you don't really know that you don't have it. So maybe there's some optimal threshold that is kind of balancing those out. And so if you're familiar with receiver operator curves, that's what we're doing here. We're plotting that true positive versus 1 minus the true negative rate. In the interest of time, I'm not going to go too much into kind of the theory here, but the idea is that the area under the curve of this graph, of this curve, tells you how good of a diagnostic it is. And so if I look at my rho value as a proposed model-based diagnostic, it's okay, certainly better than nothing at determining between no versus some inhibition. That's just because the red and orange curves aren't really that different in some of these cases. But I do much better kind of over 0.9 at determining low versus high inhibition. And so that is going to tell me something from a kind of a clinical perspective of how good my test is. We can also ask about other processes such as liver metabolism. Glucose and fructose are differentially transported to the liver and differentially metabolized by the liver. And so we might ask ourselves, well, how does the rate of the liver processing affect these curves as well? And if I were to put a tracer on just the fructose moiety of the sucrose, or just the glucose moiety of the fructose, as my collaborators did here, we can see that you get different curves for fructose and glucose. Usually fructose in yellow comes a little bit earlier. And if I were to look, it turns out that liver metabolism is also primarily associated with the Rho parameter, and not so much with the others. So that's kind of a problem. So multiple processes are reflected in the same mechanistic model parameter. It's an inherent limitation of the breath test, though. It's not a limitation of the model. It's the fact that the dynamics of the breath test look the same if you have a dysfunction in the gut versus dysfunction in the liver. And so that needs to be accounted for if you were to use this test clinically, that there are multiple signals that could be accounted for. So maybe you think about the test as a non-specific dysfunction test, and maybe as a preliminary screening for those who need further testing. And so you may ask, rightly, is it possible to disaggregate these processes? Probably. Identifiability depends on what model information is being measured. So if you were to measure more things about the system, you can get probably more parametric information. So what if you were to measure serum glucose or serum bicarbonate? And that could provide additional parametric information. The challenge, though, is that we're interested in a non-invasive method. And so we should not assume that we could have this data. And so this is a thing as me as, saying to the trialist, being like, it would be great if you could collect blood every 15 minutes. And the trialist is saying, like, you have a child that is like screaming and is so unhappy that it even has to sit there and give breath every 15 minutes. Like, you're not going to get blood. Also, there are institutional review boards that are not going to let you collect blood every 15 minutes. So I think it's helpful to kind of remember that there can be a disconnect between what we would like to do from a theoretical perspective and what is practical in the real world. So coming to a time here, just to kind of recap. So this carbon-13 sucrose breath test was able to detect EED and other gut dysfunctions in a non-invasive way. But if we just use standard summary measures, some empirical curve fitting, we have no idea if that's really connecting to specific biological processes or not. If you come up with a conceptual model for the pharmacokinetics, as have been done before, but they weren't able to make that model fit well. And the reason is that it's not identifiable. And so having somebody that has some experience with differential algebra and identifiability, we're able to come and make a reduced model. And finally, by leveraging these inhibition experiments, we're able to create a model-based diagnostic, which we then kind of give back to the IAEA. So I'll stop there. I know kind of there were a bunch of questions in the middle, but still happy to answer questions. I don't quite know how typically long this seminar goes, but I'll stop there and see if we have more questions. Thanks so much.
All right, thank you so much for your very nice talk, Andrew. All right, so do we in fact have some more questions or comments? All right, so if not, then let's...
Quick question. Yeah, so how far is this from the actual implementation and practice?
Great question. So the International Atomic Energy Agency has used this test in a set of field trials in children in 9 countries, although I think they only completed in 3. And so they're currently looking to see whether the test was kind of successfully detecting EED in those children, and I think that will kind of depend how much it's used going forward. What's nice though is that even if sucrose is not the right tracer, and there are also some indications that it may not be, because digesting sugar is a really fundamental part of human digestion, and so it's probably not one of the first things to like go when there's dysfunction, and so there's some indication that a different tracer might be worthwhile. But what's nice is that this approach that we've come up with here is, we can do our model reduction, we can do our differential algebra identifiability on a different tracer.
Thank you.
One thing that is nice is that the IAEA is including the modeling in part of their chapter to its member states in their report this year. So I feel encouraged that it is kind of seeing an audience.
You mentioned that in practice, it is hard to take samples of breath tests. And so I'd like to ask you, how often must these sucrose breath tests be repeated in order to gather enough numbers of observations of Y so that you can actually estimate the derivatives nicely?
Yes, that's a great question. One challenge with that is that the answer may depend on the dynamics themselves. So that if most of the work that we're doing is within healthy adults, we can get enough information with every 15 minutes for, say, 2 hours is probably typically enough. But if for people that have dysfunction, that peak in their breath curve happens much later. And so within 2 hours, you may not be even detecting the peak, and that may be not giving you as much information as you would like. So it's challenging to come up with like one specific number that works. So one of my graduate students is now working on some kind of very practically oriented work to give the IAEA a sense of, which summary measures and versus the model-based diagnostic are best. Can you get away with running only a 60-minute test, a 90-minute test, a 2-hour test, a 4-hour test? Do you need to measure every 15 minutes? Can it be every half an hour? Can it be 15 minutes for the first hour and then half an hour thereafter? So, so your question is a very good one. It just doesn't have an easy answer.
So how long does one injection of the C13 tracer last in terms of the number of observations that you can make?
We have tested up to 8 hours, and it's still observable above baseline levels of carbon-13 at that time. So I don't know exactly how long, but kind of beyond 8 hours, I think you're kind of losing information. But the problem is that as a diagnostician, you want to have the child there as little as possible. So the question is, how little can you get away with?
Thank you.
Thank you.
Actually, I want to thank you for a very interesting and clear talk in terms of the practicality, because last time we had a similar talk on identifiability, and the question about how to actually use the models is a little vague, but now it is much more clarified. Thank you very much.
Great. So I know that this wasn't a very theoretical talk or probably stimulating from that perspective, but hopefully it gave a sense of, the real world impact that this kind of work can have.
Right, absolutely.
Yes, thank you very much. Could you say, so what is this relation to atomic energy?
Great question. So the International Atomic Energy Association is this is a little off the record, I'll say, is that I think that their work in stable breath isotopes, they're promoting the ways in which nuclear techniques, which include stable isotopes and not just radioactive ones, can be beneficial to humanity. And so I think actually quite a bit more funding came into this space after the Fukushima nuclear disaster. And so, there are, so this nuclear techniques for nutrition is a large part of their portfolio at the moment as a way that this kind of information can be beneficial to humanity.
Thank you.
So are there still uses for exact solutions to differential equations if we can come up with them or find them? I mean,
yes. So like in this case,
yeah.
Let me go back. I think I had a slide on it. The exact solution here is useful because what people are currently using is just like fitting this either of these models to the breadth curve data and then saying, based on that curve, what's the cumulative recovery, what's the integral at 90 minutes? Well, this first curve in particular is particularly nasty from the perspective of integration because it doesn't integrate to 1, it integrates to infinity. And so, that might, for 90 minutes, maybe it's close enough, but it has some kind of unappealing traits from that perspective. So I'm hopeful, although it's hard to make paradigm shifts, that like this first curve fits just as well as these other empirical ones and is better from the perspective of having some interpretability of the parameters. So I do think there's value in the exact solution.
Thank you. Yeah.
All right, so if there's no more questions or comments, let's thank Andrew again for this really nice talk. And I'm looking forward to seeing everyone again next week.
Thanks so much for having me. Appreciate it.
Thank you, Andrew. Have a good one.
Thank you. Bye-bye.